The centrosome is also known as the microtubule organising centre, which is essentially the head of the skeleton of the cell. So it sits nestled right next to the nucleus, and it's from the centrosome where all the microtubules radiate out, where proteins are shuttled and cargo is shuttled up and down, so from the nucleus generally out to the periphery of the cell. So as proteins get made, they get shuttled around, and so the centrosome is really responsible as well in a killer T cell to direct the delivery of secretory granules to the plasma membrane. The understanding that the centrosome is essential for dictating the polarization of secretory granules from the centre of the cell to the periphery to the plasma membrane was a really landmark science paper about a decade ago from Gillian Griffiths' group in Cambridge. And they showed that actually in T cells, and it's a very, very unusual phenomenon, the microtubules, the centrosome polarises and docks right at the plasma membrane, right at the very point where the T cell receptor signalling has occurred. So it's essentially like a T cell saying a laser beam to the membrane saying, right, here's where we need to kill, here's where we need to release our cargo, throw grenades at this point. T cells receive all different levels of signals, so weak signals and strong signals, and the level of those signals can ultimately dictate cell function. So the stronger the signal is, the faster the centrosome might get recruited to the plasma membrane, and also the faster the granules get migrated down those microtubule tracks and delivered to that synaptic cleft where they can release their toxic contents into the cleft. So the strength of signal is really important to think about in terms of when we're designing receptors where we want to trigger killing. The affinity of those receptors and the avidity of the strength of interaction is ultimately important for dictating cell fate. T cells that display a higher avidity or a strength of interaction for their target will recruit their centrosome and granules more effectively to the immune synapse. So the strength of binding of a T cell to its target affects how the centrosome is recruited and how rapid cytotoxic granules are recruited. And today, as I'm recording this, on the day of recording, we've just submitted a paper for publication, so it's not out yet, so no one else knows this yet. But what I can tell you is that the CAR T cells, these engineered receptors, actually recruit their granules to the membrane faster than T cells. So, and what that means is that once that lethal hit has been delivered and the target cells decide to die, once they've undergone that decision to die, there is some sort of signal that the target cell tells the T cell, okay, you've done your job now, you can detach from me and go on and kill another one, because they're serial killers, because one T cell and one target cell doesn't make for a very efficient immune response. Now, if something goes wrong where that lethal hit doesn't get delivered, or the signal's not strong enough, or the target cell is resistant to cell death, which does happen in some cancer cells, the T cell doesn't detach rapidly. So it stays bound for a really long time. So in a normal, healthy situation, it would have done its job and released in about 4 or 5 minutes. In some cases, it can stay on for hours. And the consequence for the immune system is this T cell's still getting this danger signal from the target. And so what does it do? It starts calling for backup. So how do T cells call for backup? They start releasing pro-inflammatory cytokines to recruit other cell types in to help them do their job. And so it results in this sort of cytokine storm and cytokine release syndrome in patients with faulty or defective T cells or target cells that are resistant to cell death. So there are very important consequences for the immune system when this process of cell killing changes or even is just delayed in its kinetics. These findings are, of course, really important when we're thinking about designing cancer immunotherapies. We want to be able to design a T cell that's a serial killer, that takes out many, many targets, that kills quickly, that kills efficiently, but without all of these off-target side effects of pro-inflammatory cytokine production. You can imagine in a child's developing brain that has high-grade glioma, Cytokine production and edema and swelling inside the brain is the last thing that you need. So these are the sorts of things we're thinking about when we're designing these receptors. How can we design a receptor that will kill quickly and efficiently without these often fatal side effects?